everyone. Good morning. Welcome to the Milwaukee Public Museum's Learn with an Expert program. I'm going to give us some time to let everybody connect. Looks like most of us are going to be connecting from YouTube today. Welcome, welcome. All right, excellent. Excellent. Perfect. Okay, it looks like we have most people are going to be joining us now. So I'm going to go ahead and get started. Welcome, everybody. My name is Maisie. I am an educator here at the Milwaukee Public Museum, and I am so excited to be joined today by Dr. Javier Ortega-Hernandez. Just a quick little bit of housekeeping before we get started. This program is going to run about an hour. It is best suited for grades 6 through 12, although everybody is welcome to join. So even if you're grown-ups, you're welcome to join too. You don't have to go. So like I said, it's going to be about an hour. We will have time for questions at the end. So if you were on Zoom, that means that we are going to use that Q&A section. So we're not going to be monitoring the chat so much. Just make sure to use that Q&A section. On YouTube, looks like most of our friends are joining us from YouTube. You can log in to your YouTube account to ask questions on the chat there. So that's what we'll be doing there. All right, so without further ado, I would love to welcome Dr. Javier Ortega-Hernandez. He is the Associate Professor of Evolutionary Biology at Harvard University, and he is also the Curator of Invertebrate Fossils, and he's going to tell you all about what that means and what his research is. So I'm gonna thank him and have him join us. Thank you so much for joining us, Javier.
Cool. Thank you, Macy. It's great to be here. And thanks everyone for tuning in. It is a great pleasure to be here in the Milwaukee Public Museum to give this talk. And as Macy mentioned, I'm an invertebrate paleontologist, which I realize that this is a meet an expert. So you're technically meeting an expert on dead bugs. And you know, these are animals that are very close to my heart. I enjoy them very much. And I want to share some of the things that I find are really interesting about them with all of you, right? So my talk is entitled Breathing New Life into the Earliest Animals, and I will go into a bit more detail what that specifically means in this context. But before we get started, I have a quick announcement. So this is an interactive talk, right? You can actually interact with some of the material I'm going to be showing. And what you need to do is to take out your phone. You know, most of us will have like some type of smartphone. You want to open your camera and you want to have either Wi-Fi or, you know, be able to have access to the internet. If you scan a code like the one shown on this screen on the lower left, you will be prompted to open a link, and this link will take you to some 3D models, which are the main data that I will be talking about today. These are models produced from the fossils themselves, and this is what the fossil actually looks like. This is not a reconstruction. This is the actual organism itself as we are able to study it using the techniques that I will be talking about today, right? So with that being said, what we understand today as biodiversity, you know, it's very much similar to what we see here. We have in this slide just a few of the more endangered species in the world. But when you think about animal lives or plant life, this is very much similar to what you may have in mind. These are animals like, you know, vertebrates and fish and birds, all of these things. But the reality is that back in the Cambrian, so back in the origin of animals, things look really different. So what I do at Harvard is that my group specializes in understanding where all of this really wonderful diversity is coming from. So we want to study fossils that are going to tell us how all of these animals diversified, how they evolved, how they originated. So if we were able to take a dive into the Cambrian Ocean approximately 500 million years ago, things would actually look a little bit more like this. This is a reconstruction produced by researchers and scientists at the Royal Ontario Museum in Toronto. This is an animation produced by an artist known as Lars Fields. And what we have here is basically British Columbia, so Canada, about 505, 508 million years ago. So we're looking at a marine environment full of these really weird creatures that look nothing like the things that we have today, like this very charismatic Anomalocaris right in front of us right now. So, this is a very different world compared to what we have today. And what we want to do is to really understand how this came to be. The way that we can do this is by actually studying fossils, right? So I'm a paleontologist. I'm here to talk about fossils and to show you some really good examples. So in this case, we have really a treasure trove of Cambrian fossils from all around the world. Again, approximately 500 million years old, which is a long, long time ago. And the main thing here is that we specialize on the study of fossils which are exceptional. So in this case, these are organisms that have no shells. They have a carapace, but they are, soft by all intents and purposes. So we can only really get these really exceptional soft fossils under very peculiar environmental circumstances. And, one of the most important of these sites is actually found in South China. So if we go to the field in South China, in Yunnan province, this is what some of these localities look like. It's very lush, very green. This is what is known as the Mao Tianshan Hills in Yunnan province. But of course, we will not be able to see fossils if we go to somewhere exactly like this. So we have to go to some quarries. And this is me back in 2017 checking out some of these rocks. And what I'm showing you here is essentially what one of the localities of what is known as the Chengyang biota is known, what it looks like in the field. These are all a whole bunch of mudstones of very soft shell. And these rocks actually contain some of the most significant invertebrate fossils in the world, which are among the oldest and most important for us to understand the Cambrian explosion as this origin of animal life. So in this case, there has been a ton of work dedicated to study the paleobiology and the evolution of these organisms. There are many books that you can buy that include really wonderful fossils like this one on the right, which is a very peculiar type of worm that has some parasitic worms associated with it. And what is really interesting about Chengyang is that it is the oldest site that has this really nice quality of preservation. So it is really quite important. In addition to, having this great diversity, we also have extremely striking evidence of whole body organisms, including not only their external morphology, but also parts of their internal anatomy. So what you see on the right over here is actually the preserved remains of the brain in this really early arthropod, which is, again, really impressive. So when we take all of this information together, what we have is that Chengyang in South China are significant because they are telling us a lot of information about the early evolution of animals during the Cambrian explosion. And it can tell us this based on their morphology, how they look like, based on their modes of life, how they interact with each other, and also in terms of how they live as part of a greater community in this marine ecosystem. So this is all good, so these are all kind of like fantastic things. So what is the problem? So the problem, as an occasional reviewer may say, is that many of these fossils are really flat, right? They are essentially roadkill, as I have been heard to say at some point. And the problem here is that this is going to distort what we can tell about these organisms, because we as animals are three-dimensional we're three-dimensional beings. We live in a three-dimensional world. So if we have a fossil that is basically a pancake of itself, we are going to be very limited in how much information we can obtain from it. And this is going to make it more difficult for us to understand about early animal evolution. So what do we do? One of the great advantages of these fossils from Chengyang is that they are preserved in a very peculiar way that makes it so that they are enriched in iron. So because of that, we can actually take our fossils and put them inside a machine known as a micro-CT scanner. And this is essentially a machine that is going to produce thousands of X-ray photographs by rotating the specimen very slowly. So what you can see here is that the device on the left is the X-ray gun. The device on the right is the screen where the photograph is going to be taken, and the fossil is in the middle. So as the X-ray gun is shooting, the fossil rotates. And in doing so, this screen is going to be capturing thousands of images that are then going to be used for producing a 3D model of the morphology itself, right? So this can be actually super helpful in the same way that if you break your bone and go to the doctor, they can tell you, well, your bone is broken. We can also get a lot more information from these fossils in this way. So if we were to look at the data, this is more or less what it looks like. What I'm showing you now is that we are going through this fossil, through all the different slices that this device has captured. And what you will notice is a lot of gray, but every now and then there's some blobs of white material, like right on the top right now. That is the fossil itself. And the really cool thing about this is that we can actually, using some computer programs, we can extract that morphology and we can see this in glorious 3D, which is going to let us know a lot more about the evolution of these organisms, right? So with that in mind, I'm going to talk to you about a few case studies that really nicely demonstrate the capabilities of this approach. So the first one is trilobites or trilobite-related arthropods. So many of you may be more or less familiar with trilobites. They are very common invertebrate fossils. Most trilobites that you will be able to see or even collect if you go to a place like Utah, for example, are going to consist of the hard shell. So the trilobites are not dissimilar to a lobster in that they have a carapace that is really tough. And it is very tough because it has a mineral component in it. It's called calcite, right? So in this case, what I'm showing you here is a trilobite known as Olenoides serratus from the Middle Cambrian shell. And you will notice that it has a few extra features that we will not usually see in normal trilobites. So these fossils are really exceptional in that they have all this bunch of legs. And these are really exciting because they let us know a lot more about the ecology and the behavior of this animal. But here's the thing. Because trilobites generally have legs that look very similar to each other. They're very samey, kind of relatively boring if we are a bit mean. This has been taken as evidence that trilobites are really primitive animals. They are some of the earliest organisms that we have in the animal fossil record. And because of that, people think like, well, they're kind of silly. They're kind of primitive, not very interesting. And what we want to do is to really be critical, like, is this really the case, or is there something else about trilobites that we are missing, right? So in this case, this brings us to the first data that I want you to take a chance to scan. So just to remind you, you can take your phone, open the camera app, and then if you scan this QR code on the lower left, you will be able to see the data that I'm going to be showing you right now, but you will be able to open it in a program called Sketchfab, and you will be able to interact with it using your phone and basically rotate it in whatever way you please and see its morphology in way more detail. So please go for and experience this talk in this way. And this is one of like 6 that I'm going to show, so there will be plenty too. So in this case, I want to talk about really quickly about this organism known as Cynoburus lunaris. So Cynoburus is an arthropod known from the Shenyang biota. And this is a pretty rare animal. It is not the most rare, but it is pretty rare. There are about 10 or so specimens that have been collected over several years. And this makes it so that we used to not really understand its morphology really well. But the advantage is that, of course, we have access to this material. Before I got into that, the reason I'm bringing up Cynoburus is because, so we know trilobites, and we know trilobites have this calcitic skeleton, but trilobites are not the only arthropods that we have hanging around in the Cambrian. Trilobites have a great extended family, like all of their cousins, if you may. And the difference here is that many of these cousins do not have a mineralized shell. They are softer, right? So trilobites are hard because they have a mineral exoskeleton. These other animals do not have a mineral exoskeleton. They have a normal exoskeleton. So this makes it so that they would have they would be more similar to a cockroach in terms of how soft they are. They're still a bit crunchy, but they are not really tough, right? And because of that, we only see evidence of this species in some of these exceptional sites. In this particular case, Cynoburus belongs to this group on the right called the Sandarellids. And because of that, we only get to see it under some really special circumstances. So what we have on the left is a fossil of Cynoburius, as it looks like when we find it in the field. And, you know, it's not too bad. We can see that it has some legs, it has some segments, it has this really lovely crescent-shaped head shield. And what you see on the right is a photograph taken with ultraviolet light that allows us to have a bit more contrast between the rock, which is a mudstone, and the fossil itself. So the fossil looks really dark, and this allows us to get a little bit more information. But this is still not super good. It is better, but it is not amazing. So if we put this into the microCT scanner, we can actually see on the underside of this animal. So what I'm showing you now is the 3D model that we obtain after we put this into an X-ray scanning machine. So on the left side, we have the dorsal view, so the back view, and that would be similar to what we see when we actually grab the fossil in our hand. But then when you have on the right side, this is what we would call the ventral view or the underbelly, right? So this is where things get really interesting, because we can see this part of the fossil that would be impossible to look at if we were just going to examine it on the surface. And this is really exciting because we can see all of these lovely details about the limbs and the body and the antenna and the head. And one of the things I want you to take notice of is the scale bar over here. So the scale bar is 1 millimeter. So this means that this animal is about this big. And we can see every single segment in the legs of this animal and all the tiny articles that make the antenna, which is just remarkable. So one of the great advantages of doing this is that we can then, because this is a tomographic model, we can further explore it in our computer. So we can actually take a closer look at what these limbs are doing, and we can do a virtual dissection. So instead of taking the animal and like cutting it live, we can do the same thing with a computer. And this way we can obtain a much greater understanding of its organization. So what we see here is that we have all of these limbs. So what I'm showing you here is just like one limb. It is just shown in different orientations to see its morphology a little bit better. And what we have is that there are two parts. One part is the walking leg, which is called the endopod. And the other part is called the exopod, and that would be more like a respiratory gill. And what this is showing us is that there is a very, very long walking leg, which is almost like an antenna, and the exopod or the gill is really short. But this is only with the legs which are on the head part of the body. If we look at the heads which are on, if we look at the limbs which are on the back of the body, the morphology is actually very different. The walking leg is a little bit shorter, it's a bit more what we expect in an arthropod, and the gill is actually much more developed, right? So what we see over here is that the legs on the head and on the body are actually quite different, and this means that they're going to have different functions. So when we put this together, what this is telling us is that Cynoburius, even though it is not a trilobite, it is very closely related. And it is telling us that some of the trilobite relatives that we have in the Cambrian have very strong evidence that the limbs were not all the same. The limbs were a bit differentiated if you look at the head or if you look at the body. And this is important because one of the reasons why arthropods today are so significant and so important is because they are essentially like a Swiss Army knife. They have all of these segments and they have all of these legs, and they can basically do whatever they want with them. They become really functionally versatile. And here we are seeing some evidence of that all the way back into the early Cambrian. But Cynoburius is not the only example of this sort of very nice differentiation. We have another animal called Pygmaclicpetus. And again, we have a lovely model. So I invite you to go and use your phone to scan it if you want to do it. And Pygmaclicpetus is even more rare than Cynoburius. Pygmaclicpetus was only known from about 5 or so specimens, again, after many years of collecting. And this animal is even more tiny. This animal is about, like, it's smaller than a penny, really, or like a quarter. So it's a really, really tiny thing. Now, what I'm showing you here is a schematic of the initial description of Cynoburius, of Pygmaclicpetus. which again is not very detailed. We can see it's an arthropod. It has a head shield, it has some segments and a tail, but again, it's not super detailed. So once more, by taking some of these fossils from the Cambrian and actually giving them the good old microcity treatment, what we have is actually some really impressive information going on. So I'm showing you here what the original fossil looks like on the upper left. And then in yellow, you can see all of this really rich morphology. And very similar to the case before, we can turn it around and see the underbelly of this animal and see that it has these really dramatic limbs. All the limbs are very different. And what is super exciting about this animal is that the gills are really impressive. So one of the big advantages is that we can select specific parts of the animal and we can then remove them so that we can have a better view of some of the underlying features. So in this case, what we have done is that we have digitally removed the exoskeleton, like the upper part of the body or the carapace, so that we can see the legs in great detail. So on the left side, we have the back view, and all of these paddle-looking or finger-looking things are actually the respiratory gills, which are really, really dense. And this is going to tell us something really quite interesting about how this animal used to live and how it used to breathe oxygen from the water, right? And on the right, we have the underbelly again, and we can see some details of the mouth and the limbs and how they are all really, really different. So very similar to the case before, we can put this information together. We can do some really nifty reconstructions to show how all of the limbs in the body are very different. Like the antennae are very different from the head limbs, and those are very different from the limbs in the body. And this means that they are going to have different functions. Some of them are going to be used for sensing. Some of them are going to be used for walking or for feeding and for swimming and breathing. Again, these are arthropods, and arthropods do they take most of their life choices using their limbs, because that is what they are able to do for sensing the environment or to just making a living. One of the things which is interesting about Pygmaclepiatus is that, again, we can include this into some analysis and understand a little bit better the deep evolution of all of these features. So what you see on the right is just a tree of life, which is a result of some analysis. And what this is telling us is that actually this type of gill structure that looks like all of these paddles or lobes actually may be really, really old, and it may be ancestral for this broader group of animals that also includes trilobites. So the take-home message of this part is that Pygmaclepiatus is showing us this critical evidence that the limbs of trilobite, that the limbs in this animal were very different. And what is really exciting is that Pygmaclepiatus is actually like a trilobite granddaddy. So in a way, the fact that we see trilobites having very similar limbs is maybe not really accurate, or it is accurate, but it is actually a case of secondary simplification, whereas the early ancestors of this group were actually much more complex. So what this is telling us is that trilobites are not primitive by any stretch of the imagination. They had a really complex morphology, and they had a really complex ecology as well. It's just that we have to consider them as a whole in a broader evolutionary context of these organisms if we want to really understand what they are telling us about the early evolution of animals. But, you know, we can move on. And one of the other ways that we can use these approaches is to discover new species, right? So in the previous two cases, we were revising species that were already described based on fragmentary material, but we can also use these approaches for finding out, hey, there are new species known to science that we had no idea, and we would not know unless we actually look at the underside of these organisms. So here, there's another QR code. Please feel free to check it out. And this is a bit of a tricky story because the sketch I'm showing you here is something that is originally called Gentiania furcatus. This is a super tiny animal, also from Shenyang. The abundance is extremely rare. This animal was initially described based on only 2 specimens, and I'm going to show you them. These are the 2 specimens. So you see 4 photographs because these are the part and counterpart. When we split open a fossil, we usually have 2 pieces. So I'm showing you all of them to you. And, you know, if you are very observant, you may realize that, well, these don't really look like the same thing, right? Like the 2 photographs on the left look like something, and the two photographs on the right look like something else. There is something fishy going on. And part of the problem here is that these are also really small specimens. Again, this specimen may be about this big, so they can be really difficult to work with. And if we want to describe them based only on their superficial appearance, it can be really tough. So, you know the drill now. Once more, MicroCity to the rescue. And what we can do is actually have a much better view of what is happening on the underside of these organisms. So the one on the left is Gentiania proper. So that is the original species. But the one on the right is actually a little bit too different. It is not Gentiania at all. It has some other features. And that is what I'm going to show you right now. So in a similar way, if we turn it around and if we see the underside of this animal, what we will see is that it has these really complex, really peculiar limbs. So you can see this better on the lower right corner over here. We have a zoom in into these limbs, and they basically almost look like tentacles, but they're like really weird articulated tentacles, and each one of these limbs has a very, very strong spine. So this is really, really peculiar. And if we see at the upper right, we are looking at a part of the head, and these features that are labeled T1, T2, and T3 are actually some of the segments in the head region, but they are really, really small, very reduced compared to the rest of the body. So this is a little bit odd. But for me, when I first saw this, I thought like, oh, I know what this is. I have seen this before. And the reason is that these animals actually belong to a group of arthropods, again, from the early Cambrian of China, known as Fusinhuids. And Fusinhuids are super weird extinct arthropods. They are known exclusively from the early Cambrian of South China. They are not found anywhere else in the world, which means that they are endemic to this region. And Fusinhuids are really interesting. They're very close to my heart because I did a lot of work with them as part of my PhD. And they're really interesting. They are some of the earliest arthropod ancestors. They have a very peculiar morphology. They have way more legs than they need for some strange reason. And what you will be able to realize is that some of them also have these very reduced segments in the head, what you can see in the photograph on the lower half of the screen. And they also have these almost tentacle-like limbs. They are arthropods, and their limbs are jointed, like arthropods today are. But their limbs are like the legs of Fusinhuids are just very unique. So this is a feature that we can use for saying, aha, we know what this is like. So in this context, what we were able to do is to, from very, very little material, so this is based on one specimen, but it is one specimen that actually was preserved super well, and that we were able to get a very nice view using computer tomography. We can actually put this into its broader evolutionary context, and we have basically evidence that there are some hidden Fusinhuids that we did not know about, and we basically have to be able to come to this material and study it with new techniques if we want to get a better understanding of their extinct diversity. So what this discovery is telling us is that even fragmentary or incomplete material can yield new surprises when it is looked with the right tools, right? Which is part of the reason why this approach is so useful for us when we try to really squeeze out as much information as we can from these fossils. Moving on, we have another of these weirdo animals that we have, before we took this approach, we had very little idea of what it was. And in this case, this is an animal called Ercaecunia multinodosa. So here we have a sketch of how it was initially described, which again is not a lot to go in. It's a little bit rudimentary. And you may think, well, this sort of kind of it looks like a shrimp, maybe, if we squint a little bit. And, you would be correct. This is actually a shrimp relative, but we needed to have more evidence to actually demonstrate that that is the case, right? So again, there's a QR code, feel free to scan it. And this animal has some lovely limbs. So it is really worthwhile if you are happy to do so. Which, by the way, we'll be including also all of links to all of these models in the links below when this is published. So you can actually go and access all of these models at your own leisure later on, if that is your preference. So in this particular case, again, we have the original fossil on the left, and you can see some similarities with the sketch. It has this carapace, it has like a chunk of antenna just messing around, it has this long body. But again, this is not very detailed. So once more, we take micro-CT, we turn it around, we can see all of the limbs in this animal. And the limbs are really the important part over here. We can see that this animal had this very well-developed antenna. It has some sensorial structures, which are highlighted in blue. And what is really exciting is that we can see some jaws or some mouth parts. So those are highlighted in green, in purple and in this like other shade of purple. So that this animal has jaws and has essentially two pair of antennae is really useful because those are features that we see in crustaceans today, things like shrimps and crabs and lobsters, right? And then when we go and actually see the limbs of this animal in a bit more detail, we also find that we have a walking leg, which is highlighted in green, and they use it for moving around. We have a gill, which is highlighted in blue. But these animals also have a second type of gill, and this is what is highlighted in red. And this is a very peculiar feature called an epipodite. So epipodites are features that we see in quite a few crustaceans today. If you have never seen these features, that is very common. That is not a problem because this is actually mainly observed in the larvae of some of these crustaceans. And it can also be seen in the adult, but it can be a little bit more difficult because it is a very delicate structure. So what I'm showing you here are larvae or babies of different types of crustaceans, of brachiopods in this case. And all of the arrows are pointing to these tiny like blobs on the legs, and those are the epipodites. In this case, because we have in Archaeocunia evidence for having two pairs of antennae, having mandibles, and having these epipodites, all of this is going to be very clear morphological evidence that this animal is indeed related to crustaceans, things like shrimps and crabs, and more significantly, that all of these animals actually had their origins all the way back into the Cambrian explosion, again, 500 million years ago. This is something that we already had a bit of a good idea of. There are some techniques that allow us to extrapolate when animals originated, but always having this direct evidence from the morphology is going to be very helpful for our purposes. The last thing I want to talk about is this really funny animal that we published a study in this animal about a month ago. And this is an animal called Isoxis. So Isoxis is very unlike anything that we have today. We have some crustaceans that have a similar looking carapace that like swim in the water column all day long. But Isoxis is really special because it is one of the most distant ancestors of arthropods that we have. And, you know, you can tell it is an arthropod because it has a shell. It has these really cute, big, googly eyes. And the limbs on the front clearly have articulations, right? They have these raptorial limbs that they would use for grabbing prey. But the problem with Isoxis is that this animal is known from many localities around the world. Mainly the carapaces are known. The carapaces have a very distinctive shape and they're very easy to identify. But the problem is that we very rarely get the limbs. And the other problem is that because they have this really beautiful carapace, the limbs are usually covered, which makes our life real difficult. So you know what to do. We have to micro-CT scan it. In this case, I don't have a QR code for this one. I apologize because it didn't scan that terribly well for the purposes of that. But we are still able to have some pretty impressive fossils, and we still have some really great tomographic data. One of the reasons why Isoxis is so interesting is because it's telling us something about the earliest stages of arthropod evolution. And thanks to new material, again, from the Chengyang biota in South China, we actually have evidence that in some cases, the carapace is going to be a little bit off because of decay, because of disarticulation. The carapace will be a little bit out of place, and this will allow us to see the body in greater detail than we could ever before. So what we can see here is that Isoxis, unlike other arthropods, has a soft body, which is really peculiar, and it is very similar to some of the even more distant ancestors of arthropods. So, you know, Isoxis is a weird, soft arthropod with a carapace. But what about the limbs? That is what we really want to know, because usually we have this pesky carapace getting in the way. what are these limbs doing, and what can we learn from them for early arthropod evolution? So the good news is that, again, isoxis is very amenable to micro-CT scanning, and it requires a little bit more work, but it is actually possible to isolate each one of these limbs and to really see their structure in way more detail. So what do we have on the left are just two different isoxis specimens that have been scanned, like the full body. And we can see some of the details of the limbs, and the one on the lower left actually has some details of the gills as well, which are suspiciously similar to those of Pygmaclipeatus, which is really interesting, and there may be a broader story there. But then this video on the right, which is spinning around, is one of the limbs, one of the walking legs that this animal has, or more like a swimming leg, because these animals were mainly swimmers. And what we can see is that there are very clear segmental boundaries along the length of the animal. There are a few holes, and the reason for that is because when these animals are replicated by iron, the iron doesn't always go through all the spaces. So sometimes we can have some holes in between, but we get a pretty good idea of what the morphology looks like. So what this is telling us is that isoxis gives us really nice evidence that these jointed legs that arthropods have, which are very distinctive of most species that we have today, evolved before having the hardened plates on the body itself. So this is telling us that becoming an arthropod is not just like it's not like you clap and now you're an arthropod. Actually, this takes a lot of time, and evolution happens very gradually. So first we acquire the legs, and then we will acquire the arthropod-like body, which is really fascinating. So just to conclude, what I hope to have done in this talk is to show you how in all of these fantastic specimens, we can always get a bit more data if we have the right tools, right? So computed tomography, or micro-CT for short, is a really powerful tool that allows us to study Chen-Yang fossils in great detail, because we have this understanding of how they became fossilized. And this way, we can basically leverage the technology that we have available for obtaining new information and basically learning more about their early evolution. This gives us new insights into their morphology, their ecology, how they live, and broadly speaking, how they are actually going to be informing the early evolution of arthropods, which are the most diverse, and it's one of the most ecologically important groups of animals that we have living today. So we really want to understand where all of this extant or modern biodiversity comes from. And in this case, some of these early fossils from the Cambrian are giving us this story little by little. We have to be reconstructing all of this very complex history by studying different groups of organisms, but this is going to come together into a single narrative that allows us to get a much better view of what life was like during the Cambrian. In a more technical term, micro-CT is really amenable to processing a lot of samples. One of the great advantages, and something I didn't really mention until now, is that usually when we have to work with fossils, it is not uncommon that they have to be prepared. And with prepared, I mean that they have to have the rock removed from them so that we can see them in greater clarity. This can be very helpful because we can reveal new features, but it can also be really tricky because if people are not super careful, you can destroy the fossil, and that is no good, of course. So in this case, because computed tomography employs X-rays, it is actually non-destructive and it is non-invasive, which means that we can scan these fossils as many times as we want, and they are never going to be affected. They are not going to be degraded in any way. This is fantastic because we can preserve these fossils in museum collections for a much longer time and still be able to make the most out of their morphology without having to destroy them. And finally, one of the great advantages that this approach has is that I am able to do what I have just done with you, in the sense that people can be more involved in really interacting with all of this data. It is possible to have different teaching and learning experiences, because it is not the same that I'm just sitting here and just telling you all about these fossils. because now you have the chance to actually interact directly with this model. So there is a really great opportunity for making learning more accessible and more exciting by using some of these emerging technologies in order to share this with all of you. So with all of that being said, I just want to give a quick thanks to my collaborators and co-authors, both in China and in North America, that have been really wonderful to work with, as well as some funding sources. Funding is also very useful to have around. And with that being said, I just want to leave you with this last one model. This is an animal I did not have a chance to talk about, but this is, I think, my favorite model. Like, if you're going to scan one thing today, this is the one that you have to do, because it's really, it came out amazingly. It's a very small specimen, but we have really lovely data in this one organism. And which is really fun is that even though this fossil is known from like over 100 specimens, we were still able to get that little bit of extra juicy information that is telling us that little bit more about their early evolution. With all that being said, thank you so much for joining in, and I am very happy to answer any questions that you may have.
Awesome. Thank you so much, Javier, for such a really interesting presentation. I was sitting there at my computer trying to scan everything. So we're really, really excited to be able to have some time for him to be able to answer your questions. So I see that we have some questions already. I'm going to give us a little bit more time to get those questions in. And I'm going to get us started by asking my own question. Which is, so Javier, you are from Mexico, and we were talking before, you've been all over the place with your research. And I'd love to hear a little bit about where your research has taken you.
Of course. So as I mentioned before, I'm originally from Mexico City. As part of my education and getting all of this experience to really be and training to become a scientist, I have lived in the UK for 10 years. I moved into the USA about 4.5 years ago. But one of the great and exciting things about this line of work is that I am able to travel all around the world for research, for conferences, for collaborations, for making new friends. So a lot of my work takes me to different parts of North America. So next week, I'm going to Utah to do some field work, for example. We have been in Canada, we have been in Morocco, in China, in different parts of Europe, in Australia. So, depending on the year, it can be very, very variable, but it's always really exciting. And one of the great things is that it is possible to do a lot of this work in collaboration with colleagues and scientists all around the world. And together, we are able to produce something that is bigger than the sum of its parts, right? Because we are relying on different kinds of expertise, different access to material. And some of this work would simply not be possible without some of these really excellent relationships that we're able to build. So, it's just one of the perks of being able to do this on a global scale.
That is really, really great. Being able to have all of those experiences in so many different places sounds really, really special. So it looks like we have quite a few questions in YouTube. So I'm going to actually start because you mentioned going into the field. And so Samantha has a question asking, like, how often are you in the field, like digging in the field versus doing this type of imaging work?
Mm-hmm. So we will try to go to the field for research purposes at least 1, possibly 2 times a year, and we would be there more often. But we need to do teaching, we need to do some administration, so we usually go during the summer. But when we do, we try to make it worth our while. So we usually try to go for 10 days, 2 weeks, something similar to that. Because we also have to apply for permits. We have to work a lot with local communities in order to obtain permits for doing some of this field work or getting in touch with local collaborators, of course. And when we're not in the field, we are also doing a lot of work which is based on museum specimens. In a way, going to museums, like here the Milwaukee Public Museum, is almost as good or sometimes even better than going to the field, because museums are really absolutely full with specimens that have not been looked at in years. And what you need is to have someone with fresh eyes or with just like a little bit of a different background or expertise to be able to say, hey, this is something interesting. Let's look at it in a bit more detail. So between fieldwork and museums, we actually spend, quite a bit of time during the year engaging in those activities.
I love, as a museum educator, I love that you plugged the importance of museums too, because I think a lot of times people, they see the exhibits and they think that's the entire purpose of a museum, and that's really only part of it. And part of it is providing access to researchers as well. So thank you for plugging museums. There are quite a few questions about trilobites specifically. And one person mentioned that trilobites are Wisconsin state fossil. You learn about that in school. And so someone was wondering, like, are there kind of regional differences? Like, do we know anything about the differences between trilobites here in Wisconsin versus trilobites somewhere else? Were they kind of the same?
Yeah, for sure. So that's an excellent question, and really it's insightful. So I showed you a lot of this evidence of exceptional fossils, like all of these soft-bodied animals, and they are telling us something about the Cambrian explosion, right? But actually, the way that the Cambrian explosion, or one of the ways that it was initially recognized as this major evolutionary event, was because of trilobites. And that is because when we go back in the past, 500 million years ago, the continents had a very different organization to what we have today. So if you can imagine North America, so basically Mexico, the USA, and Canada, and a little bit of Greenland as well, not being where it is today, but basically being completely tilted on its side, and then basically close to the equator, that is where North America was back in the Cambrian. And then most of the other continents were basically put together into this supercontinent called Gondwana. China was divided into like North China and South China was like in a whole other place. And because of all of these geographical differences, different groups of trilobites evolved and occupied each one of these different continents. So in a similar way, why we have, for example, kangaroos in Australia and they're nowhere else, or the reason why there are no bears in Africa, for example. So we have these trilobites that were very unique to North America, some trilobites that were very unique to South China or to Australia or to South America as well. And this happens really soon after we have the first evidence of trilobites. So the first trilobites already show that they have become diversified and they have different flavors in the different countries that you visit them, right? So the only way that this could be explained is by assuming, well, how did this happen? I mean, usually, migrations take time and evolution takes time. So surely there had to be a really rapid event of evolution at the beginning of the Cambrian that could explain why the trilobites in North America, in Australia, and in South China are so different from each other. So there are differences. And basically, the further back you go in time, the more emphasized those differences become.
That is very, very cool to hear, like how quickly that happened too. Very neat. I'm going to keep going with trilobites because we have two other questions about trilobites, specifically about their legs. So one question is, what could they have used the leg spines for? And the other one is, do we know if their legs could regrow?
Oh, wow. Excellent. So what are the spines in the legs for? The legs of trilobites have very, very close to their body, so right underneath their body. The way that trilobites feed is that their mouth would not be facing forward; it would be facing back and on the underside of the body. So what trilobites do when they feed is that they are going to sit on top of whatever they were feeding. They were going to move their legs, and they have these very, very spiny parts of the legs called anathobases, which basically just means like the feeding teeth. And when these are moving, they are able to shred all of these food items and then transport it forward towards the posterior facing mouth. So this is just a very peculiar way in that they would be feeding, but the spines are really critical because they are basically like their teeth. They have no mouths in the way that we have them. They have no jaws, but they still have mouth parts. So they use their legs for feeding and for transporting all of this food towards the mouth opening. Now, what is interesting is that trilobites could feed of different kinds of organisms. So some of them could only feed of very soft worms or very soft organic matter. And some of them were able to feed on animals that had a hard shell, not unlike crabs today. So some crabs can feed on snails, some crabs can only feed on like very soft worms, for example. And regarding the second question, whether they could regrow their legs, so yes and no. So yes, we know that there are arthropods, and all arthropods basically can regrow their legs if you pluck them, right? Arthropods grow in this way in this mechanism called molting, in that they will produce a new exoskeleton when they have like the right size. And if you remove the leg of an arthropod today, when they molt, they will regenerate that leg, which is really neat, right? Now, this is difficult to prove for a fact in trilobites because trilobites have never really been discovered to have evidence for a regrowing leg. But we know that there are trilobite fossils that were bitten. So you have your trilobite and it has like a chunk taken off, like if you grab a cookie and then you take a chunk. And in this case, we know that they survive because they actually show some of the spines that are growing back, like on the exoskeleton. So this tells us that trilobites were definitely able to regenerate. Even though we don't have evidence of their legs regenerating, we have reasons to believe, based on our understanding of arthropod biology, that they very likely did regenerate their legs.
That is so cool. And I would love to see a fossil of a trilobite with like a chunk bitten out of it. That's so neat.
Oh, absolutely.
Okay. We have a question from Stefan that asks, what is the earliest examples of antenna that you have encountered? I did see, a lot of these little guys have antenna. What are the earliest examples of that?
Yeah. So this is a question that can be answered in like different ways, because we can ask, what is the earliest example, as in the oldest example? Or what is the earliest and what is the most primitive or the most ancestral animal that has antenna? So the oldest example would be basically the trilobites from the locality that I'm talking about here, the Chengyang. So we know that trilobites, when they have sufficient preservation quality, they will have limbs. And all trilobites that have been discovered to date with limbs have antenna, all of them, right? So in a way, similarly to our understanding of their biology, suggests that even the earliest trilobites, which are a little bit older than the ones I talked about today, will have antenna, even though we do not have evidence of them in the fossils, but we know that their slightly younger relatives do have antenna. Just for context, so the fossils I was talking about today are from about 518 million years old, and the absolutely earliest trilobites are just a few million years older than that, about 520, 21. And the problem is that these first trilobite species, which are known from North America, from Morocco, and from Siberia, are preserved a little bit not too well. We have their carapaces, but they are a bit cruddy. Because of that, we do not have evidence of the limbs. But again, they're arthropods, and many arthropods have antenna. So it is a safe bet to assume that these early trilobites would have also antenna. But then in terms of their evolution, we have many groups of animals that actually have antenna around the Cambrian explosion, and that would come from these sites of exceptional preservation. So trilobites and their relatives would be some of the earliest animals that have antenna. But for example, these Fusinhuids, these very weird looking animals with really tentacle-like limbs, they also have antenna. So having antenna is just like a good idea because they allow us to sense your environment. So it is a very common adaptation for arthropods back in the day.
That is super cool. Very, very neat. Let's see, what other questions I'm trying to think of the ones that I want to ask in order. Okay, so there's another question in the Zoom Q&A here, asking if there's any recent discoveries that have like changed the way we understand arthropod evolution.
Oof, yes.
Like, how much time do you have?
Yeah, I mean, you can come and take my class. And even that would not be enough. One of the really exciting aspects of this is that there are so many groups of people around the world working on understanding early arthropod evolution and early animal evolution, broadly speaking. So every year there are discoveries that like really change what we know about these animals. Some of them I showed you today, like for example, Soxys being able to show that it had this soft body and this kind of like jointed legs. That's kind of like useful to know. I didn't really talk about this in this presentation, but the discovery of nervous systems in Cambrian arthropods has also been really fundamental in how we understand their early evolution and the limits of exceptional preservation. We have new species of even more ancestral arthropod ancestors that tell us that their ecologies were very complex and very different in some ways to what we have today, but also very eerily similar to other groups of organisms. So yeah, I mean, part of the reason is that the Cambrian was such a dynamic time, and we only are looking at very small snippets of information at any given point in time. So new discoveries are going to be refining our view of this period. Not all the time, because of course, like a new species of this or that is not necessarily going to be groundbreaking in of itself. But sometimes we have these animals with this very unique combination of traits or structures, and those are going to be really interesting when we try to contextualize this into their broader evolutionary tree.
Right. Very cool. All right. We have two, actually more than, but I'll focus on two right now. Two questions kind of about your favorites. So one from Zoom, one from YouTube. So Devin on YouTube is wondering if you have a favorite fossil, and Stefan on Zoom is wondering if you have a favorite place to dig or like do field work.
Okay. I'm really bad with these questions because I always take them too seriously. But I mean, I think, yeah, Fuxing Hu, it's like these weird ones with the long legs. They do have a special place in my heart. These are the first fossils from China that I had the opportunity to work with. And for me, that was really important because, I mean, I was a third-year grad student, going on my own to China for the first time. And, I was hosted by like wonderful collaborators. And it was a real like life-affirming experience. Not only from the fossil perspective, which was, again, these are like gorgeous things to look at all day on no end, but also from the connections that I was able to make and being able to just keep working with all of these colleagues after more than a decade by this point. So, it's not just because of the fossils themselves, but also all the extra baggage that they have with them, that they are, they're really special to me. And then in terms of the fieldwork, yeah, I mean, basically the more distant the better, to be honest. I mean, again, so for example, when I have gone to China, it's not really to do fieldwork, it's more to work on museum collections. But that is always a great time because it's such a, cultural immersion. The same with Morocco. Going to Morocco to do work with colleagues or to do field work or to go to conferences is always fantastic because it's just such a wonderful country. But yeah, I mean, basically, every place has its own quirks and its own hidden gems. So, for example, we went to British Columbia, to like really, really far up north, Canada, a few years ago. And that was great because we were in the middle of nowhere. So you get like this really great immersion with nature and just trying to avoid the bears. So every one of those experiences is really quite something. So, they're all special in their own different ways.
And I actually really love too that you point out that yes, of course, the research and the fossils are really important, but those connections are really important too. I think a lot of times some people think of scientists as being very, they think of us as very serious and but it's, those are very important as well, as making those connections and also making that research.
Yeah, absolutely.
All right, so we are right at time. I'm going to end with the last question on YouTube. I'm sorry, it's another favorite one, so apologies. And maybe you've already answered it, but that last question on YouTube, Samuel is wondering, what is your favorite part of your job?
Oof. I think that the sense of discovery is really, it's really quite something. Like I call it like getting, it's almost like a science high. Like sometimes, like this is something that I don't get to do like as much, because now, my role is to support other people to make sure that they can go into the science, right? So one of the things I enjoy the most is like the aspect of like going to the field and doing the thing and like really getting into the weeds of it. And that's something I still do, maybe a little bit less now, but it is really, it really kind of like fills my heart whenever like a member of the lab comes and says like, hey, I have this really cool data to show you. And it's like, that is really cool data. So, it's really exciting. Because you are seeing something for the first time, like you're like the first person, or in my case, the second person, because they are the first person that actually saw this. You're one of the first people that actually sees like this really new insight into how the world works, or how like the deep history of the planet. And, that sense of discovery, I think, is one of the reasons why a lot of people like fossils and like people like paleontology. And, if you go to the field, and it's like a tiny treasure hunt in a way, like if you go to the field and you crack open a rock and there's a fossil there, like you are literally the first person in history that has seen that fossil in that moment, right? And being able to get that sense and to be able to contextualize that into, oh, I know what this means for our understanding of animal evolution. It's just like a real satisfaction, and it's a real privilege to be able to do that, because it's very rare to be able to say like, oh, there's a new discovery to be made. So that is definitely something that is kind of very much worth cherishing in the middle of like other other activities, which may be like paperwork and whatnot. So you hold on to those senses of discovery, like, oh man, it's worth it.
I think that is a truly perfect note to end on. So I'm going to thank everybody so much. Thank you to my YouTube watchers and to my Zoom watchers. Thank you so much for participating today. I hope you learned something. I hope you had fun. This is our last Learn with an Expert series for this school year. We do plan to have other experts for next school year as well. So keep an eye out for news about that for next school year. And in the meantime, I again really want to thank Dr. Javier Ortega-Hernandez for being so awesome and giving us such an interesting presentation, and giving us some really interesting like little fossil tidbits too. I can't wait to see a picture of a trilobite fossil getting with that little, with the munch out of it. So thank you so much, everybody. We look forward to seeing you all next school year. Thanks, everyone. Bye-bye.